good morning. Hello, everyone. Today we are in the FMA UPC for another session of SIMBa. And we have from London, I think, Cristóbal Rodero Gómez from King's College. And he will talk to us about modeling cardiac function shape. So one part of mathematics which has real applications. Thank you very much.
Thank you. Okay, thank you for the introduction and thank you for the invitation. So the idea that I had, let me know if you hear me well, by the way. It's okay. Can you hear me well? Yeah, okay. Okay, so the idea that I had for this talk was to talk to you about the main paper of my thesis and also give a sort of introduction to the area of computational cardiology. It's sometimes called computational modeling. So the idea of computational cardiology is that we have a model. It can be a simulation. It can be just the anatomical model that is running on a computer or on a supercomputer. And the idea is to try to input as many data as possible. Usually this involves anatomy, so the shape, the physics, so to know how the different forces are interacting among them, and the physiology, so to know the underlying biophysical processes, and then integrate it somehow in a multi-scale and multi-physics manner. This means that we are going to be modeling usually at different levels, so from a cell level to a whole organ level, and multi-physics because, so in this talk I will focus on electrophysiology and mechanics, but then we can also do about fluid dynamics, for example, and the idea is to be able to go through all these physics, coupling them also somehow. And then the output of this can be many different things. So the one that I will talk about today is this paper that you can see here, where the idea was to try to link somehow the shape, the anatomical features with the function. So to see how small changes in anatomy affect the function, but in a controlled manner, because we can do this with the simulations, and this will be completely unfeasible in experimental setups. So just some brief words about who's working on this. So there are a lot of different types of engineers. I want to highlight here software engineers, because the idea of this is not only doing research, but making also reusable and reproducible research. So the idea is that it's likely that someone else will use your code. It's likely that someone else will try to look at the data that you use in previous papers. So at some point, it can get really messy to try to understand what did you do on a script that you did like a year ago even. A lot of computer scientists, because most of these simulations, we run them in high-performance computers. So these can be very expensive simulations, and for that we need on the order of hundreds of cores. Physicists, mathematicians, as myself, of course, mainly applied mathematicians and statisticians for obvious reasons. And then there are physiologists and clinicians. So this is a bit of an area that we are usually not so much exposed to, this more clinical side, but they are really an essential part of this arena of this research, because they are the ones that first they give us the data, they tell us which problems are important to them. That is usually not the same as the ones that are interesting to us sometimes. And then once we do the research, the main conclusions and the main takeaways, what does it mean for them? And what does it mean in general for the patients? Because that's the end goal, even if it's long-term of this research. So as I mentioned, we'll talk about this paper. Just a brief warning. So I'm not going to be, I will mention all the maths that are underlying several of the processes, but this is translational research. So the idea of this is to use already developed methods and already shown results, apply them to real data. So there will be, at least that I spotted, there are inconsistencies in notation, and there are some things that you will just have to believe me that it works. Right, so with that, as I mentioned, the idea is to close the gap between anatomy and function. And there are some medical images techniques that really do that. So if you or some relative or some friend of yours ever had a scan, a medical scan, it's probably had like an MRI or a CT. And in several modalities of these scans, you can see both the motion in the case of the CT. In the MRI, you can see the flow velocities and the displacement as a vector field. In echocardiography, so this is basically the same as an echography, but just pointing at the heart. It's not very clear here, but you can usually see the anatomy of the heart as well as the displacement of several parts of the heart. So clinicians can see the anatomy and the function, in this case, the motion, at the same time. The problem is that it's not in a controlled way. So if we start asking questions like what happens if this part here gets thicker, it's not really clear what will happen to the function. So the first step is to try to quantify all these features. So to quantify cardiac anatomy, we use statistical shape analysis, and to quantify cardiac function, we use simulations, electrophysiology and mechanic simulations. And then the link here will be global sensitivity analysis. I believe that this was applied firstly in geology simulations, I believe. But the idea is that it's a sensitivity analysis that takes into account the whole space. And since for that we need a lot of simulations, then we will also need to borrow some tools from emulators. So I will go during the talk, I will walk through all these different steps. Starting on cardiac anatomy. So before I can start, I need to do a brief recap on the biology of the heart. So the parts that we're interested here are the chambers. So the bottom chambers, the ventricles, are the ones that pump the blood out of the body, and the atrium act as reservoirs while the ventricles are pumping to hold the blood there. The left one, that it's usually on the right side in a very confusing way, pumps to the body, while the right side pumps to the lungs. Then we have some veins on top that are bringing the blood to the heart, and also kind of holding the heart in place, as well as the pericardium, that it's basically like a bag of tissue that is surrounding the heart, and it's also keeping it in place so it doesn't move too much towards the chest. This is how it looks in a textbook image. This is how it looks on a CT scan. So here it's a bit more difficult to understand which part is which. So the first step in this is always segmentation. So segmentation, basically, it's a fancy painting. So we have, so these are images, and we have each image for each slice. Usually it's, the distance is under 1 millimeter. So the idea is that we have to paint each one of these slices, the part that is the heart. We don't do this manually. This would be like unbearable, unreliable, very time consuming. So we're using some networks to do that. And then those networks usually gave us the main chambers, so where the blood is. But for the simulations, we're going to need the walls. So what we do is usually dilate those blood pools with a fixed thickness, and that creates the walls. We use for this, so for this study, we use 20 healthy patients. So these were patients, these were subjects or people that went to the emergency room with some chest pains. They did some analysis and they didn't find anything cardiac related. So they are classified as healthy or as symptomatic at least. We did all this in the software Sec3D. It's an open access software, and the idea is to use some Python scripts to try to automatize as much as possible. This is how it looks in 3D. So this is just the segmentation, like stack up one on top of the other. So besides the main chambers of the heart, we have the aorta here in yellow, the pulmonary artery here. And then we needed some auxiliary structures for the simulations, meaning this vein rings is just a label to know where the vein will be, because that will act as boundary conditions in the system of equations. And then we have some planes here closing, because to be able to track the volume, we need to have a closed volume for each chamber. So we need to artificially close it to be able to see the changes in volume and then the changes in pressure. Once we have the segmentation, we mesh it using CCAL. So this is a library based on C, and the idea is just to, since we are going to run the simulations using finite element methods, we're just using tetrahedra and linear elements. It's there's probably a smarter and faster or more stable way to do it, but it's the most reproducible way so far, especially with these really complex geometries. So we mesh it with tetrahedras to see, and they have an average edge length of 1 millimeter, and we are talking in the order of 2 to 3 million of tetrahedra. We will also need fibers. So the heart is a muscle, and as almost every muscle, it has fibers. So the fibers will be where the electricity conducts faster and where the contraction follows the direction. So we can define angles both in the epicardium, so on the outside of the heart, and on the endocardium, so on the inside. And then we can make this direction of the fibers to rotate. So basically, these are just vectors in the mesh for each point that tell us the direction of the fiber. So I talked shortly about this pipeline, but this was basically the first year of my PhD to make one of these hearts. So then we applied this pipeline to the data set, and we end up with the cohort that you can see in the screen. We did some quality checks. So the main value to quantify the quality of the mesh is the scale Jacobian. So basically, if we have a perfectly regular tetrahedron, the value of the scale Jacobian is 1. If it's completely collapsed, it's 0. And we had an average of between 0.7 0.8 for all the meshes. So we were quite happy with that. So that's from the computational point of view. And then from the clinical point of view, we need also to check that the meshes they look like hearts, basically. So one of the checks that we did was to compare the volumes of each one of the chambers of the ventricle and the atria with literature values, and we see also that it fits within the expected ranges. And then we made these available, so they are open. You can just go to the link and download it. They are VTK files. I'm not sure if you are used to that format, but it's the most common in these kinds of applications. So basically, you have the coordinates of the points, and then for each triangle, it tells you which points are connected. And so far, it's been widely used. So mainly for to test algorithms to use as benchmark for educational purposes. So instead of having a regular grid as square to show how the heat equation propagates, for example, it's been tested like on these hearts to show in a more realistic geometry. So now we have the cardiac anatomy, but now we need to quantify changes on this. So the idea is to use this technique called statistical shape analysis or statistical shape models. So this model, this field, the idea is to get some sort of average of the sample of the hearts, and then we can see each sample how much how different it is from this average. So then we can see how abnormal it is from this template. So we have some sample shapes. The classic way was to select some landmarks, some important points in the mesh, and then literally Euclidean distance and take the average and see how those differ using some principal component analysis, for example. The problem with this is that our meshes look like this. So it's not really clear which landmarks should we use. And the other option is to use all the points, but it can get really expensive. And also that we will need the exact same number of points for all the different cases, which is not the case. We could do that, but then we would lose some quality in the anatomy. So then we wanted to look something that it's not depending on the number of points, that is not dependent on the meshing technique. Here's where the DRAM currents come. And this was a bit of an unexpected application from calculus and from physics. So the main idea of the DRAM currents is if we think of the mesh as a set of points, the idea is to go to the dual space and work with a set of vectors and a set of mappings. And then since we're working with mappings and not with points, then we do not depend on the specific points anymore. And then we can work a bit more on the doing statistics there. So to go to this dual space, the idea is that we need to provide it with some structure to be able to use statistics on it. So the idea of this comes from Faraday's law. That's why it's called currents. So the idea was from this core idea that the electricity and magnetism are the two sides of the same coin. So using Faraday's law, we can link currents through a coil with the electromagnetic field inducted by it. And the idea here is to, in a way, think it as running many currents in this coil, and running many, and that will generate a lot of magnetic fields. And so if we have those two informations, then if we have enough information about that, we can also retrieve the shape of the coil. Only that the coil, in our case, it's the mesh. So we want to know the shape of the mesh in this setup. So to do that, we will need basically the magnetic fields are vector fields, and the currents will be scalar numbers. So the idea is that each surface then will be characterized by a set of numbers that are based on vector fields. So the idea now is to how do we define the space where these vector fields live, and we need a space that has some nice properties. So then we can go to the dual space, and then we can also provide some metrics in the dual space to be able to compute the statistics. So yeah. So I won't go again too much into detail for the sake of time, because I want to go a bit through all the paper. But the idea is that we can define this space as the closed span of a kernel times a vector. That this space called momenta for historical reasons, because this used to be used in physics. So it has to do something with velocity, I guess. And this kernel, it's mimicking in a way normal distributions, because then it has some really nice properties statistically. And it is that there's this parameter here that is called the window width. And in a way, if we think about fitting a histogram using a normal like curve, small values will catch too much detail. So it might be noise. So we want to have as enough detail, but not too much. So this will be one of the parameters that we will need to choose. So then we can define a current just as the linear mapping from the space generating by this to the real numbers. And this is how it looks like, different choices of this window width. So if we, so here we are having some like two curves, two segments, and then we are visualizing the vector field, so the current of the difference of these two curves. So you can see that if the window width is too big, then we don't capture much detail. So then this doesn't really follow the different curves. Well, if we use a small enough detail, then we can really go through the different lines and go from one to the other. That's the idea, right? So then we can move to the dual space. We can get a base of the dual space. Again, I won't go much into this. This is one of the steps that you will have to believe me. And then once we have the basis, we have a scalar product, and we have an inner product, then we can get a norm. It's not actually norm, it's more like a similarity metric. So we are not measuring how far the currents are, so the surfaces are. It's more how close they are. So then our algorithm will go to try to make them as close as possible in the terms of the similarity metric. So yeah, as I mentioned, now there are problems on where do we choose these points here and where do we choose the window width. So the idea for the I will show it later, but the idea for these points is to select them in the center of the triangles, and then the beta number will be how big the area of the triangle is. So even if that then depends on the specific discretization of the mesh, then there are some results that I am sure the convergence, as the mesh gets finer, then we'll get unique and convergent result. Then we need a way to deform one mesh into another. Since we're going to measure the shape of the meshes, we also need to have an average mesh, and then we also need to be to modify one mesh into another ideally in a continuous and smooth way. So here's where this technique called large deformation diffeomorphic metric mapping comes into play. So the idea is that if phi here is the transformation that we're looking for, and the bar is the template, so the average shape, we choose one of the hearts as the initial one, and usually the initial transformation is just the identity. So we measure how far it is, and then we have some regularization technique to avoid overfitting. And so the idea is then use the transport equation to find which is the shortest path from each one of the samples to the template, or the other way around, where this regularization technique is called velocity. Again, that doesn't have to do anything with velocity in our case, but for because it started in physics. And this depends again in another kernel, like the one that I showed before, that it looked like the Gaussian distribution, where the points here will be the control points. So these control points will be a set of points that are distributed in the space, so not on the mesh, but on the space itself. So then when we apply this transformation, it will be these points that move, and this will drag, in a way, the mesh. So let me show you first before going to the control points. Since we might have the hearts in different positions for the different patients in different coordinates, we first align them. So we just chose the middle point of the valves, and also the lowest point in the wall between the left ventricle and the right ventricle, and then we just align it as best as possible. And this is what I meant with the control points. So in this toy example, the control points are the intersection of the grid, so the corners of the square. And as you can see, as we deform one shape into the initial shape into the final one, what moves is not really the points of the line, it's more the space itself. So that's why that's a way to visualize also the dual space. So we are not in the points of the specific shape, but on the space where that shape is embedded. In our case, the control points look like this, so it's just uniformly distributed in 3D. So then these points will move, and that will drag the heart with it. Right, so this is how the template or the average heart looks like. This was from the 20 meshes. Actually, it was 19 because we discarded one of them because it had one more vein here. So that was a difference in topology, and for the diffeomorphic deformation mapping, it needs to be the same topology. Again, here I'm using topology in the engineering sense, not in the math sense, so patience. And then since we end up with too many points, because we end up with the number of, we can end up with a matrix of the number of columns be the number of hearts, and the number of points is 3 times, because we're in 3D, the number of points in the mesh. It's too many points really to analyze and to do any statistics, so we need to do some dimensionality reduction. So we use principal component analysis. It's actually singular value decomposition because it's not square matrix, but they call it PCA everywhere. And the idea, if you're not familiar with this technique, the idea is to find a change of coordinates so that we can match the maximum variability in the data, and then we can just get rid of some of the dimensions. So in this case, we can just stick and describe this this data with only one direction instead of two. As I mentioned, we discarded one heart because there was too much variability in the left atrium. And so then we can describe, once we do PCA, we can describe the heart as a linear combination of a set of vectors. And since these vectors are common for all the hearts, we can describe each heart as a set of numbers, as these eigenvalues. I will usually refer to eigenvalues or eigenvector as modes. This can be confusing at the beginning, but by the context, I think it will be pretty clear when are we talking about vectors and when are we talking about scalars. We discarded the last one because it explained too small of a variance, so less than the precision of the machine. So we can sort the variance explained by each of the eigenvalues, but we can sort them by the variance that they explain. So then the first modes explain more variance, and we can discard the last ones I mentioned. And then we can just choose a set of modes that describe the shape good enough. So we choose 90% of the cumulative variance to be explained by the PCA. So this is just 9 modes. So then we can describe, having the template and having the transformation mapping, then we can describe each heart just with 9 numbers, really. So then we need to check how they look. We need to make sure that they make sense. So you can see that mode 1, see in this video also better. Mode 1 basically is the shape, is the tilting of the heart. It goes like more forward or more back. Mode 2 is the global size, so if it gets bigger or smaller, but also a bit of the thickness of the wall. And then mode 3 is more like the torsion. So we can try to visualize this with all the modes. It gets more messy as we go more into smaller modes. For example, mode 9 is just a thickening on the left ventricle, so like around this part. So the idea is that the later the modes, then the less variance they explain anatomically. So now we explained, we are able now to quantify cardiac anatomy. We know by this set of modes how the heart is. So now the idea is to go to cardiac function, to go to cardiac simulations. So for the electrophysiology simulations, we use the reaction iconal model. The idea is kind of like the reaction diffusion equation, but instead of the diffusion part, we have the iconal that it looks like the one on the screen, where basically we have we just want to know when the wave reaches each point in the mesh, where V here is tensor characterizing the conduction velocity. So basically, the higher these numbers, the faster will go the electricity in the fiber direction, in the cross fiber direction, so the sheet direction. And so we have the normal one, so more towards the inside of the heart. So that's why we needed the fibers here, where the fiber information comes into place. And this is how a simulation looks like, a screenshot of the simulation. So the right ventricle, the wall is way thinner than the right ventricle. So the wave reaches first on the outside in here compared to in the left one. And then we also compare the activation of the heart with the clinical measurements reported in the literature. And we saw that also fitted quite nicely. And then we have a lot of parameters that we don't know about. So we have the conduction velocities, and we also have where do we apply the initial conditions. So for the initial conditions, I will show you later, but basically we applied a region in the endocardium that is known where it's usually the electrical wave starting. And then for the conduction velocities, we fix some parameters from the literature, because the idea here is to have all the parameters the same in all the hearts. So then the only thing that is changing is the shape. So then we can see how the effect of the shape affects these simulations. Then for the mechanics, first we need to, so when the heart is electrically activated, then the cells contract as soon as they activated and they overcome certain threshold. So to do this activation, we need to couple the electrophysiology model with the mechanics. So this is how it looks like the active tension equation. So basically the way to couple the time that a node is activated with the tension that is generated. This doesn't have any meaning really. This is phenomenological, meaning that the people who developed it did some experiments and they measured some curves, and they saw that this equation is able to capture all the relevant information of these curves. Then we need to model the passive stress. So the idea is to then how to model the formation of the tissue. So this is not my expertise really, so I just know the basics of it. But the idea is to then even if a part of the heart is not electrically activated, like the aorta, for example, if the part underneath moves down, then the aorta should move down. That's the idea. So then we use the Guccini model. So this was a model for hyperelastic materials, and the important bit here is this exponential here. So basically, at the beginning, it can deform a lot, but then it gets really hard. And for the parts that are not the ventricles, it's been shown that this Neo-Hookean model works quite well. For example, for the aorta or for the pulmonary artery, it worked quite nice. And this is a mix between physics-based and more experimental-based. So this is how the simulations looks like. So in yellow, you can see the electrical activation going over, and as soon as we have the electrical activation, the tissue contracted. And then we can put it in real time, and then we can see how this moves down. There are some things that I didn't mention that is usually the hardest part of the simulations, that are the initial and the boundary conditions. So this region here is where we apply the initial conditions for the electrophysiology. And then for the boundary conditions, for the mechanics, the boundary conditions are twofold. So first we have the pressure in the endocardium. So we need to establish an endocardial pressure. Sometimes we have the measurements from that specific patient. But in this case, we use literature measurements again to fix all the functional parameters to have all the difference in anatomy. And then we also need to somehow fix it in space. So we use spring boundary conditions in the right pulmonary veins and the superior vena cava. So here we know that this happens in the heart. And then in the pericardium. So this bag of tissue that I mentioned in the beginning, we also put it because we saw that the motion was artificial. So this part was coming up instead of this bit coming down, because then we can modify the stiffness of these springs. So if we put stiff springs here, then we kind of fix the tip here. So then that forces this part to move down as it should. And then we don't have a circulation model, but we need a way to track the pressures. So there's this 0D model called Winkesel model that is basically mimicking electricity modeling, where instead of electricity resistance, we have resistance to flow, so the valves, and instead of capacitance, we have the changes in volume. Right, so once we have the simulations working and the anatomy described with the modes, then we can link it, and the idea is to use global sensitivity analysis. So this is a type of variance-based sensitivity analysis. And in any sensitivity analysis, the idea is to see how changes in the input affect the output of whichever function. Usually in a local sensitivity analysis, we move a bit the input, and then we see how that affects the output. But the idea here is to see how changing all the parameters affect the variance of the output rather than the specific change. So yeah. So if we have a model here, this is the expression of the sensitivity analysis first order effects. So that is that we fix a specific parameter to a specific value, then we change all the rest doing many evaluations of this function. So in our case, the function is the simulation. Then we take the expected value. We repeat this with the same parameter, but to a different value. Repeat all these steps several times, as many as possible. So that will give us, then we can compute the variance, and then we just normalize over the total variance. So then these first order effects will vary between 0 and 1. So if it's 1, it means that changes in that input parameter is what is driving the variance in the results. If it's close to 0, it means that it doesn't have really that much effect. So the idea is that now we'll see our input will be the different modes. So because we can create new hearts and we can check the modes of the hearts that we have. So these eigenvalues of the PCA. Then we can see how changes in those input affect the output. Then in our case will be the simulation, several metrics there from the simulations. The problem here is that I use the word many twice. So that means that there are a lot of simulations, a lot of evaluations, and the electrophysiology not so much, but the mechanics for each simulation, it took around 8 hours in 500 cores, so it's not really cheap. So we need a way to substitute that or to replace or to deal with it. So the way to deal with it is with Gaussian processes. So basically, a Gaussian process is an extension. It's an stochastic process. So it's an extension of a function where instead of for each input we have one output, for each input we have a distribution. And then with this we can do stuff as we do with regular functions like regression, for example. So in our case, the observed data as in this GIF will be the simulations, and then we will try to emulate the simulations. The nice thing about this is that we also have a measurement of the variance between it. Since it's a stochastic process, when it's a distribution, we can measure the some metrics from those distributions. So for each point, we have a variance and standard deviation. So then we can tell how confident are we about that that emulator. Yeah, so the idea for an emulator is basically a linear regression. So this is just a polynomial, and here we have a Gaussian process with zero mean. So then we make sure that for the training points, so for the simulations, we have it goes exactly through that point and it has no variance, and then we just add some Gaussian noise to avoid overfitting. I mentioned training points because this is usually considered a machine learning algorithm. It can be considered an optimization algorithm, really, where here the parameters will be all these hyperparameters for the Gaussian process, for the noise, and for the interpolation bit. So moving a bit forward, as I mentioned, the idea is to have our input will be the different weights of the modes, so the different eigenvalues for each heart. Then we'll do a simulation. We'll have one output, which could be, for example, changes in volume or changes in pressure, where we will use the actual simulations to train an emulator. So then we will have one emulator for each output, and then we will repeat this many times. So then we can do a variance-based sensitivity analysis, or GSA, where we will show modes 1 to 9 in this way, and then modes 10 to 18, since we usually didn't have much effect, we just group them in the same chunk. And then multifactorial is when, for example, mode 1 and mode 2 don't have much effect on their own, but when they interact with each other, they do. So finally, this is one of the last slides. And there's a lot of information in this slide, but the one that I want you to focus on is two things. So the blue ones are volume-based outputs. So for example, when the heart contracts, which is the volume of the chamber when it's contracted, or how much it contracted with respect to the resting state. So we see that it's mainly dark red. So it means that the first modes are the ones that are driving this, which makes sense because the first modes were the ones that were more difference in variance, in anatomical variance. But when we go to pressure based phenotypes, like in this one, we see that is mode 9. So this was the thickening that I was showing you before. There was a small change in anatomy, but as you can see, it can cause a really big change in the variance of the output. There are some limitations to the study as always. So the cohort size, we're using 19 hearts. Statistically could be better, but we have to think that this is data from real patients. This is from hospitals, so it's not that trivial to find this kind of data especially for healthy people. And then validation. So this is validation is a common step in this kind of studies, where the idea is to try to see how well does your model or models predict. And since we don't have other patient-specific data besides the anatomy, we cannot really do validation. So the only validation that we did was compare it with measurements in the literature. We also just measure the impacts of the modes, so the impact of anatomical parameters. The next step will be to check the impact of functional parameters. Also, we don't have any flow or valve modeling. It's just simplified models. And we contracted the ventricles, not so much the atria, because since we don't have flow modeling, we don't have a full venous return, so it doesn't make sense to contract the atria either. But we place them there to have realistic boundary conditions. And so the main takeaways of this study is the ones that I mentioned before, that the small changes in anatomy can have a big change in function, such as this one. Also that for different outputs, it matters different parts of the anatomy, so big changes or small changes. And this can have an impact on the imaging community. So meaning that, for example, for some techniques such as echocardiography, the resolution is not that nice. So it's very likely that we cannot capture small changes like the one in the mode 9. So then if we are going to use simulations based on that image, it's very likely that we are not capturing a proper distribution of this kind of output values. And we made these meshes, as I mentioned, open access, and we also generated more so extreme cases of the PCA. And we also generated 1,000 more just because we could, really. So then it can be used also for like machine learning algorithms, for example, to have a big sample of realistically looking hearts. And just a final slide on the takeaways of not the study, but of the talk. So we are not the first one of doing this. We use software already done, so for the segmentation, for the emulators, for the changes, so the statistical shape model, so for the simulations. So the idea is to use things that it's already done to do it and not focus so much into the development, but to be able to use it and maybe develop the parts that we need for our case, for our study. Also, we are not the only one. So I've been fortunate enough to be in 2 teams because I had 2 supervisors during my PhD. So it's really nice to not do this alone. So I'm not sure if you noticed, but in the paper, there were close to, I believe, 10 to 15 authors. And it's because this is not a study that could be done with one person. So this is also a way to thank those teams. And also we won't be the last ones, meaning that we need to make the code as nice as possible. So we have the philosophy in the group is then to always publish the code on GitHub, made any available data also accessible to Zenodo. And if we develop anything nice enough, like this, for example, for the emulators was from a colleague during the PhD that started developing some specific stuff for the heart. So then he made it available and uploaded to pip. So then it was available as a standard Python package. And with that, that's all. Just a quick mention that if you are looking for any funded PhD position, or if you know anyone who's looking for it or a postdoc, we have positions available to just either check the Twitter account or send me an email. And just let me know what are you interested in. And yeah, for sure we can find something that it makes everyone happy. Thank you.
Okay, so thank you very much. If anyone has any questions, please come up. There's a mic here that should be working, I think. Are you hearing us? No. Oh, I thought.
And now, Cristóbal?
Yeah.
Okay, so if anyone has a question, please come up and ask it. Cristóbal, one question.
Yeah.
How will this continue? Do you have anything planned already?
Yeah, so there are several ramifications of this usually. So now I'm working with a different data set, but using basically the same pipeline, the same methods, but to a different study. So the one that I'm working on is for heart failure patients. So these were patients that their heart wasn't pumping properly, and it was so then they were taking some drugs, they got better. And the problem is that they are better as long as they keep taking the drugs. But these are people like as young as like 30 years old, 20 something. So it's not ideal to take drugs for life that young. So then they did this clinical trial to remove them to some of them, and then they show like 50% of them improve and the other didn't really. So it's not really clear why some of them are and why not. So they just apply this kind of pipeline to see if we find anything. So to see if maybe like some change in parameters, if for some patients there's some parameter that has more effect or some specific shape feature that is really different, and then we see any there. And the other one is to see the impact of functional parameters. So in the limitations, one of the ones that I mentioned is to see how changes in the parameters of the simulations that we fixed in this study, see how that changes. So we're also working on modifying those, but that requires a lot of more simulations. So the main bottleneck there is to make them fast enough and make the pipeline as reliable and reproducible as possible.
Okay, great. If anyone has any other question, if not, let's thank the speaker again.